Good evening. Yes, audio input. Good evening. All right, you can hear me. Ne? Good evening, everyone. Komada, Komada, Jivite. I see. Yeah. Good evening, everyone. Noha says good evening. So, how is life? Heavy raining, ne? Heavy raining today. Did you go to school? How is schooling? Anything special? Good for now. Manuja says good for now. Right. It'll calm, the outside. It rained while ago. Now things silent. Life is very good, sir. Yeah, it has to be. Ne? So up and down, and then it continues. Ne? So your exams over. You didn't have any exams, ne? Not your batch. Right. So people will go with the quick discussion on the BioEve paper. Hope you have. Sir, regarding extra class, Vajra sir is keeping the Monday evening. Can you please keep it on Monday? Yeah, I'm planning to take it on Monday morning. We'll discuss this on coming Saturday. Okay, I can take Monday morning. I'm preparing the tute so that I can start it on Monday morning. Monday morning, what time we can start? What time are we going to start? Shall we start at 8 o'clock? Okay, neither. We'll start at 8 o'clock and we'll go till 1 o'clock. 8 to 1. Awesome, no? You're going to love that. Anytime, do. You stay at IMS, right? Stay with us at IMS, right? We'll take care of you, right? Keep companion. And maybe go to the canteen, eat, enjoy life. 1 to 5, no? So I'll have 8 to... No, he's not taking 1 to 5. No, dear. He's taking from 2 to 5. 2 to 5:30, 2 to 6:30, I think. 2 to 6:30. So I'm going till 8 to 1. Vajra says 2 to 6:30, as he said. No, not 1:30, dear. 2 o'clock. 2, 2, 2, 2, 2. He said 2 for me. And he want me to do the class till 1 o'clock for your bed. So then you'll have 1 hour break, right? And you can... when Monday, yes. 25th Monday. Coming Monday. Coming Monday, yes. 1:30. Oh, it's for us said 1:30. No, he said 2 o'clock. So he said that he's going to finish the morning class by 1:30. 1:30. Yes, 25 batch. So that class from 8 to 1:30, I think. Then your class he will start. 2 o'clock, ne? 2 o'clock. So we'll have the class 8 to 1. 8 to 1. Okay. And I'm going to discuss evolution part, unit 3. We'll start a new lesson. We'll go with photosynthesis, biochemistry part, as usual in the weekend class, right? I know that you love to go with that lesson in weekend class, and then we'll try to go with some new lessons. Please understand, right? Please understand, these taking extra classes is extra work, right? And tell your parents, right? This is extra work because some parents curse us. Some parents curse us. I have the voices, right? When taking extra classes, they curse one of the parent, right? Big blame for Vajra sir for doing the class on election day. I think parents must understand, right? We are not now dealing with your grade 5 scholarship exam, right? And as teachers for working for the last 20 years, 24 years, myself and Vajra sir, we have been teaching, right, for a long time. We know, right, the content and then the time management, right? Your parents are not going to, the same parents are going to blame us, nay, when we don't finish the syllabus in time also. So it's not that, neither, it's not a rose bed, the path, right, tough path, right, there are up and down. Sometimes we have to slow down, but we can't stop, right? We can't stop. We have to go continuously. So when it is the Vesak Poe Day, Poson Poe Day, Aurudu Day, what else people? Ramazan, whatever, right? Hajji, and then we have now, you have to count all these things, right? We are like, if you are doing a government job, right, with y'all, then for Poe days, Vesak, Poson, all the Poe days, right? We are not going to teach you bank holidays are there, eh? And then finally, maybe you want, right? There is no responsibility, eh? Right? Who takes the responsibility, right? So let them know, teach your parents, please, right? Otherwise, they are coming and fighting with us. Teach your parents and teach them who we are and what we do. And so discuss your matters with them. Otherwise, they think that we are just taking classes on election day, right? And that's so unfair. These are civilians. You are like the, what do you call, you have rights and they are fighting for your rights. It's not going to work, right? So extra classes. 4 classes every Monday, 8 to 1, right? Whether you come or not, we are going with that classes. Right, people? Okay, we'll go with the discussion. I hope you have the paper now. The problem is students are not talking. Don't you meet your parents? Huh? Don't you ever meet your parents, people, to talk about your difficulties, right? So don't spread wrong information. So then we have classes till 6:30, right? It will be hard to go home at that time. You talk with that with Vajras, okay? I think he's going to take the class till 5:30, as I said. As he said to me, 5:30, I don't know. Talk with him on Saturday. You will meet him on Saturday. So talk with him and fix his time. I'm taking from morning 8 to 1 o'clock, right? 8 to 1. 4 classes. How many hours then? How many hours you need a break, neither? So 8, 9, 10, 8, 9, 10, 8, 9, 10, 10:30. Break 10:30, 11 o'clock, 11, 12, 1. Ayyo. Yeah. We'll go 8 to 10:30, 2.5 hours. Break, 11 o'clock, 1. Finish, 1 interval. Okay. So you said it's Tuesday. No, I didn't say that. I didn't say any date. I didn't confirm, ne? I didn't confirm. I said that I'm going to confirm you. Now I'm confirming. I said that I have to check with the classrooms, right? People, without further chatting, then we'll go with the discussion. Find that paper, BioEve 14. Hope you have the answered the paper by now. Answer kara neda. This is right. This one. Right, and then also keep in mind, people, no extra charges for that. Okay, so tell that to your parents also, right? Some parents are going to die, right? Right, saying that we are, right, unfair in charging. I don't think so. Okay. Which of the following cannot be considered as the importance of studying biology? Can you answer, people, up to the 6th one? Can you mark the answers? Let's see whether it tallies with the answer that I have marked. And just go on marking it, please. Oops, sorry. Okay. I'm using red color. You use some other colors, students. Right. First question, the answer is second one. Which of the following cannot be considered as importance of studying biology? We'll take people second one. Studying all living species separately. We don't do that, ne? So many species are there. We can't study them all. What is the number we said, people? 10 to 100 million species, ne? So we can't achieve that. Second one, factor which is not involved in variation among living organism. Size, shape, size correct, shape correct, cellular organization correct, form not only varying among living organisms, distribution, habitat, size, shape, cellular organization, form. People, cellular organization, we didn't say that, ne? Distribution is habitat, I think, ne? Habitat. So, cellular organization, unicellular, multicellular, that's form, right? That we took as form. So, what is the answer? You go for distribution, right? It says form and habitat. So, unicellular, multicellular comes under the title form. So, I'm taking distribution, habitat and distribution. Unicellular organization, habitat. Okay, there is a issue with this question, right? Cell organization is not given, distribution is not given directly as words. Instead of cell organization, what is the word given? Form. Instead of distribution, what is the word given, people? Habitat. So if you have mark either third or fifth, I'll consider. I don't think we have anything to argue there. Nothing to argue. So either third or fifth, right? So we can't go for a final answer, right? Next one, people. Which of the following characteristic of living organisms perform their biological activities efficiently? Important, order and organization. I'm taking first one as the answer. If you have any issues, you can send me a chat. And then question number 4. So metabolism, which of the following character is important for living organisms perform their biological activities efficiently? Now we said that the order and the organization is now resulting complex arrangement which is making the living processes efficient. Okay, Nuha. So it's not order and it is not metabolism, it is order and organization according to the note. So I'm taking that as the answer. Question number 4. Select the correct statement among the elements found in earth crust. About 30% to 50% elements constitute living matter. Wrong, neither. Wrong, ne? We didn't say something like that. Human have 25 essential elements. That is correct. Second is okay, ne? Iodine is required by all forms of life. No. Trace elements found in 0.01% or less than that in dry weight of the body mass. Trace elements are found. Now, thing is, the resource book does not give any cutoff value for the trace elements, so we can't agree with that. Then chlorine is a trace element in human. Chlorine is a trace element. What do you say, people? Mucosine may be fecal. Mucosine may be fecal. right? And then in that lesson, trace element, we said minute, ne? Present in minute quantities. So this is doubtful, people, because you are going to learn this later. For the plant nutrition and animal nutrition, once again, I think we can agree simply with the statement. We can agree simply with the statement, second one, right? Now, trace and what do you call minute, minute. I gave you a list, proper list. Remember, mo, ku, zin, man, be, fe, kal, ko, crow, see, say, sen, VIF. But this list is given in a different way in the resource book, right? Later they have given this list, right? But in the first lesson, it's given as minute. So you learn the definition, right? We discuss what that is, right? Some elements are available in small amount, right? What is the difference between trace and the minute element? Now, trace element is based on this cutoff value. Trace elements we take based on the cutoff value. Minute means it's not a particular cutoff value. Some elements are present in large amount, some are present in less amount. You check your note, okay? Please check your note. Don't ask this question, right? Please check your note, or else you have to come for the Monday night class and then learn them properly. I have given the videos even. I check randa. Then I right? I can't describe it from the beginning, right? We discuss it already, right? You check your note, check your note. I'm going with the second one as the answer. Question number 5. Question number 5, select the correct statement about carbohydrates. Select the correct statement about carbohydrate. Now you all becoming again the elementary school people, right? I have to now go and teach again the chemical basis. No. You read the note, right? So select the correct statement about carbohydrate. What is your answer, people, for that carbohydrate question? Carbohydrate. Correct statement. There are water soluble polysaccharides available. That is correct. We know inulin is water soluble. Galactose is a ketose. Wrong. Water soluble polysaccharide, yes. Inulin is a polymer made of pentose. No. All branch polysaccharides can be identified by iodine test. All branch polysaccharides, no. Wrong. Ne? Cellulose. Glycogen, no. And then reducing disaccharides can be identified by the biurate test. No. So we have the answer, second one for question number 5. Question number 6. What is the answer? Select the correct relationship between organic compounds and its function. Cutin, formation of exoskeleton of arthropoda. Cutin, formation of exoskeleton of arthropoda. Cutin, chitin. Wrong, neither. And stored component of chlorophyll steroid. No. Wrong, ne? Casinogen storage protein in milk. Casinogen, storage material in milk. Correct. Glycogen, a casinogen. Is it given as casinogen or casein? Storage protein in milk. Glycogen, casein, not casinogen. Ne? So we'll take it, I mean, all syllabus, they have given it like that. Glycogen stored food in fungi, correct. And then phospholipids maintain the fluidity of the cell wall. Phospholipid maintain the fluidity. Who's actually maintaining fluidity, people? Cholesterol, cholesterol, wrong. So we have the answer, fourth one. Answer is fourth. Right, I'm moving down. Let me erase your, what do you call this? Bali, your balis. Let me erase that. All right, fresh page. I want you to mark from 7 to 10. Can you? 7 to 10. All right, done. Question number 7, select the correct statement about nucleic acid. Pyrimidine of DNA are adenine and thymine. Pyrimidine of DNA. Sir, no, sir, that is phospholipid involving in maintaining the fluidity, but they have given it as cell wall. Is it? Phospholipid cell wall kilatinone. Yeah. Phospholipid fluidity of the cell membrane. Lipid molecules are maintaining fluidity. Yeah. Cholesterol, they have given something. Cholesterol give flexibility. Shall we check that? Cell membrane says functions of the cell membrane. Cholesterol molecules randomly integrated. The cholesterol molecules provide rigidity and stability to the membrane. So I'm wrong. Phospholipids and two-side membrane rigidity. Phospholipids are amphipathic molecules. Yeah, you're correct. Anyway, we can't change the answer. Yes. Okay, it's rigidity and stability. Ne? Okay. Right, next one. Question number 7. Select the correct statement about the nucleic acid. Pyrimidine. Pyrimidine of DNA are adenine and thymine. Pyrimidine. Adenine and thymine. Pyrimidine, purine and pyrimidine. What is that, people? Purine. Adenine, guanine, purine. First is wrong. All have self-replication ability. All have self-replication ability. All have self-replication ability. Correct statement regarding nucleic acid? No. RNA can't replicate by itself. DNA form RNA. DNA can form DNA. There are no phosphate groups in nucleoside. Nucleoside is the nitrogenous base and the sugar. That is correct, people. And RNA form the cytoplasm of the eukaryotes. RNA form in the cytoplasm of the eukaryote. Wrong, neither. RNA form inside the nucleus of the eukaryotes. Now that is correct for prokaryotes. FAD is a nucleotide. No, dear, FAD is a dinucleotide. We got the answer. Third one, I agree with you. Right, question number 8. Select the correct answer with the characteristics that can be observed only in the cellular organization where fungi is included. A, no formation of spindle. B, have 80 S ribosomes. C, some can fix nitrogen. And D, have intracellular flagella, have naked DNA. Now they are asking the correct statement. Select the correct answer with the characteristic that can be observed only in the cellular organization where fungi is included. Fungi are eukaryotes. And then what features? Formation of spindle, you can see present no formation. No formation, then you can't take that. Have 80 S ribosomes, correct. Some can fix nitrogen, no. Have intracellular flagella, correct. Have naked DNA, no. So we have the answer 5. Answer is 5. Question number 9. Not a function of protein in the plasma membrane involved in the identification of the cell. Identification, correct. Give stability to the membrane. No, we said cholesterol, right, people? Act as a receptor molecule. Act as a receptor, correct. Help in maintaining shape of the cell, correct. Act as enzymes, correct. Answer is second one. Question number 10. Not a function of following cell organelle. Storage of calcium ions. Cell organelle. Function of the following. What is this cell organelle? This is RER. Oh, it can be SCR, neither. RER or SCR. SCR is actually available there. RER, the rough part, ne? Right. So, if you take people, this is function, synthesis of glycoprotein, synthesis of its own membrane, phospholipids, synthesis of phospholipids, production of transport vesicles done as a function, phospholipid, not a function of the following cell organelle, right? Uh, Not a function. Phospholipid synthesis. We say that acting as a membrane factory, right, people? Acting as a membrane factory. So, what is your answer? Following organelle. Is this RER or SCR? Synthesis of phospholipids. Now, there is a doubt neither. What is that? What they have given here? RER or SCR? I can see SCR is available too. Fine. So, a small issue. Fine. Looks like it has that. No. No, what I see is the peripheral part is SCR, right? And then the some part is going to be RER. It's both present there. So I don't think we have an answer there because we discussed the RER function, membrane factory, production of synthesis of membrane phospholipids and all. So we'll just leave that with the question. Fine. Leave that with the question, doubt. So calcium is the answer, I guess. It was mentioned in the storage of calcium. If it is not given, we'll go for that. Storing calcium ions given. Storing calcium, stores calcium ions. So we can't take that. Right, we'll leave that. No proper answer. I'm moving to next part. Right, I want you to mark the answer up to 14. Can you mark up to 14, Lamai? Right, question number 11. Question number 11. Select the incorrect statement about AB organelles. A is chloroplast, B is mitochondria. Then they're asking incorrect one. What is your answer, people? Smooth outer and inner membranes. A has smooth outer and inner membrane, correct. B has enzyme needed for the Krebs cycle, correct. Photosynthesis are found in internal membrane system of A, correct. A has stock particles, wrong. And the cristae of B has enzymes needed for the electron transport chain, correct. So I'm taking fourth one as the answer. Question number 12. Which of the following structures has the 9+0 microtubule? I'm taking following structures has 9+0 microtubule arrangement. Yeah, that's correct. Question number 10, there is an issue, no, so no point of further discussing because the diagram is not clearly showing whether it is RER or SCR, right? So we'll simply take both. right? We'll just skip that, right? So I'm taking people 12th question, which is the following structure has the 9+0 microtubule arrangement, first and fourth, right? I agree with you because the basal body has 9+0 structure too. Question number 13, group of cell organelles maintaining all major biological molecules, nucleus, ribosome, no DNA, right? So I don't think we can agree with the first one. Endoplasmic reticulum, no DNA, right? I think the best answer is fifth one. Why? Mitochondria, nucleus, chloroplast, DNA, RNA, protein, lipid, everything present. Fifth one is the answer. Question number 14. Select the correct statement about the extracellular matrix of animal cells. Correct statement about the extracellular matrix of the animal cells. Collagen is one of the glycoprotein found in the extracellular matrix. That is correct. Only collagen is embedded in the matrix of the proteoglycan. Collagen in a matrix. Some proteins are there too, right? Collagen connects with transmembrane. Collagen connects with transmembrane protein via fibronectin. Collagen connects with transmembrane fibronectin, correct? And then there is no connection of the extracellular matrix. There is no connection of the extracellular matrix with the cellular structure. Wrong. Only collagen is embedded in the matrix of proteoglycan. Then next one, people, extracellular matrix has different types of lipids that are secreted by the cell. Extracellular matrix has different types of lipids. Extracellular matrix has different types of lipids that are secreted by the cell. Wrong, that is also wrong. Second one, only collagen is embedded in the matrix of the proteoglycan. Matrix of proteoglycan. Collagen is the only glycoprotein found in the extracellular matrix. Only glycoprotein. No, we said that collagen is a glycoprotein, right? Yeah, they say most abundant. Right? Yeah, so it's only is wrong. Then the collagen connects with the transmembrane protein. That's correct. Third one. Third is better. Right. Moving people to next set. Question number 15 to 18. Right. 15 to 18. Can you mark? So is it integral protein? No, there is a fibronectin, integrin. Two different types of proteins, right? Especially fibronectin, no? Fibronectin. Transmembrane protein is integrin, right, people? Check the note. Right? Integrin, fibronectin attached to integrin. That is how it is given. Please check. Question number 15. Select the incorrect statement. Select the incorrect statement. Space formation of chromatin by using DNA and histone protein. Space formation of chromatin by using DNA and histone protein. They are asking incorrect one. So this is okay. Prophase connection of chromatids of one chromosome at centromere by kinetochrome. No, neither. Wrong. Neither. Why? Prometapase. Propase chromatin appears as chromosomes. Correct. G2-pase replication of centrosome. Correct. Anapase shortening of the kinetochore spindle fibers. Correct. So we have the answer. Second one. Question number 16. The function of synaptimal complex. The function of the synaptimal complex is tight attachment of sister chromatids at the centromere. Wrong, ne? Separation of sister chromatids at the correct time. No. Connection of the chromosome with the spindle. No. Connection of chromosome, connection of kinetochore with chromatids. No. Attachment of homologous chromosomes with each other. Correct. Answer is the 5. Question number 17. 17 following events are included in meiosis. Select the correct order. Arrangement of tetrads of chromosomes. Tetrad resource book, they have not given this word tetrad. What is tetrad? Homologous chromosome pair, right? Can you highlight the word tetrad and write there? Homologous chromosome pair we call as the tetrad. Homologous chromosome pair, right? Is a tetrad. So, all right. So, following events, you have to find people the correct order. So, what is the first one? Synapses. Collection of haploid chromosomes. Collection of haploid complete set chromosome each pole. Formation of cleavage furrow. No. So, we'll take second is A. Then next one is so we have the answer, ne? Fifth one, fifth one, right, people? BACED. BAC. Formation of two haploid daughter cells, neither. So, cleavage furrow. Once it is completed, then only you get the cells. So, therefore, that has to come last. Question number 18, what is your answer? I think I agree with your answer. Dust, not a function. Digest worn out organelles. Correct. Involved in nitrogen metabolism. Autolysis. Transport residue materials out of the cell by exocytosis. Digest food particles received by phagocytosis. So, second one is the answer. Second is the answer. Right, people, we are moving to next page now. Question number 19 to 22, can you answer them? 19 to 22, lamai marker under 19 to 22. 19 to 22 marker. Right, question number 19. Sir, BioEve 13 not uploaded to the website yet, right? I didn't check that. I'll check it after the class, okay? 13th one. I think I uploaded. Let me check. What is the answer for 17th and 18th? I'll show you the answers once again. Question number 19, select the correct statement about plasma membrane. Two sides of the membrane may differ in composition and function. Correct. Some protein molecules in the plasma membrane interact with specific biochemical molecules. The fluidity of the plasma membrane is regulated by the cholesterol. Fluidity, the stability, rigidity. Peripheral proteins are tightly bound to the inner surface of the membrane. No. Not tightly, loosely bound. Several permeability, selective permeability is an important for regulation of the exchange of materials. Selective permeability is important for regulation of exchange of materials. So how about A, B, E as the answer? Question number 20, incorrect statement about endoplasmic reticulum. Rough endoplasmic reticulum consists of tubular sacs. No. Smooth endoplasmic reticulum synthesize only lipids and steroids. How about it too? Wrong. SCR does not. SCR does detoxification. Correct. Proteins synthesized by ribosomes are sent to lumen of RER. There are membrane-bounded enzymes in SCR. So AB, wrong. Question number 21, correct statement about cell junctions. Correct statement about cell junctions. They are asking correct ones, neither. Plasma membrane of neighboring cells are very tightly pressed against each other via adhesion junctions. Plasma membrane of the neighboring cells are very tightly pressed by adhesion. What do you call tight junctions? Wrong. Plant cells do not have cell junctions. Wrong. They have. Then desmosomes mechanically attach cytoskeleton of adjoining cells by intermediate filaments. Muscle tissue has desmosomes. Human embryo has gap. C, D, E. Question 22. Question 22. Correct statement about the structure of the cell wall. Extracellular component E. Extracellular component. Correct. Correct statement about the structure of the cell wall. Extracellular component. Yes. Middle lamella is made up of glycoprotein. No. Some protista have cell walls. Correct. And then secondary cell wall is deposited on the inside surface of the primary cell wall. Correct. Cell wall becomes water impermeable due to deposition of cellulose. No. So A, C, D. A, C, D. Right. Let me show you the answers. Some of the students ask. So let me show them. Right. All right. Now we're going to do structured part. All right. Okay. Structured part 1. What is the structure given in the following diagram? This is a nucleotide. Nucleotide. right? Nucleotide. I don't think you write in detail what that is, the type of the nucleotide. No need. What is the group C, phosphate group? Phosphate group. Give two biopolymers with group C. Two biopolymers with group C. What do you write? DNA and RNA. Don't write phospholipid because phospholipids are not biopolymers. What is nucleoside? Nitrogen bases and pentose sugar. Nitrogen base and pentose sugar. We'll say that a molecule with nitrogen base and pentose sugar. Molecule with nitrogen base and pentose sugar. State how polynucleotides form in the form of nucleotides. How polynucleotides form from the nucleotides. You write little long answer. State how polynucleotide form from nucleotides. You have to say condensation between OH group of phosphate, OH group of phosphate with the OH group of phosphate of one nucleotide with the OH group attached to the third carbon atom. State two main functions of DNA. You can write them. Store and transmit genetic information. Store and transmit genetic information. What else you can write, people? Nucleus function. What else you wrote? Provide genetic information for protein synthesis or store genetic information for protein synthesis. Store genetic information for protein synthesis. Then give base pair rule. Now you write this in words. What are you going to write? Adenine pair with thymine. Adenine pair with thymine by forming two hydrogen bonds. And guanine pair with cytosine by forming three hydrogen bonds, right? You write it in full words, huh? And next one is identifying following structures. What is the structure, people? This is the tRNA molecule. make mark with an arrow the point of attachment of the amino acid. Where the amino acid attachable to the 3' end. So show it like that. And then make a mark anticodon. Mark anticodon. Okay, fine. Going to last page. Lamai, shall we go to last page? Right, next one. State the function of biomolecule transport amino acid to the site of protein synthesis. Transport amino acids to the site of protein synthesis. What is this site of protein synthesis? Ribosome. Ne? Part B1. Identify X shown in the above diagram. What is X, people? X can be the cytoskeleton. Ne? Cytoskeleton. Name structures labeled as A to C. What is A? Microfilament. What is B? B, intermediate filament or microtubule? Microtubule. Ne? Microtubule. What is C? Intermediate filament. Name the main protein types of above ABC. What are the protein types? Well, A, actin. Actin protein. B, tubulin. What is C? Keratin. Ne? Keratin protein. Mention subcellular components shown above involving in following functions. Cleavage for a formation. What is involved, people? Microfilaments mainly. I think it's not given in the resource book. Ne? Can't remember like this information in resource book. So C has a hollow and ring like nah, that's wrong. Ne? B is the microtubule. Harid. So you're wrong. No, B is the microtubule. B, I took it as B microtubule, no, correct me? Right? Why? No, not C, wrong. Formation of nuclear annulum. What are you going to write, Will? Intermediate filaments. Ne? Then next one, attachment of chromosomes. Microtubules. Mention structural difference between A and B. What is A and B? Mention a structural difference between A and B. You have to write a structural difference between the microfilament and microtubule. Microfilament, it's a filament, the no cavity. The B is a tube-like structure, right? B tube-like. A filament. That makes a difference. If you want, you can say that people the structural material too, but I think this is better. And then people, part C. Part C, what is meant by resolution power of a microscope? What do you call resolution power? Minimum distance that should present between two points that can be distinguished. Minimum distance that should present between 2 points. Points that can be distinguished. That can be distinguished. Next one, what is the reason to limitation of resolution power of light microscope? Wavelength. Ne? Mention 2 different types of electron microscopes. Scanning. And transmission. Mention 3 differences of specimen. Observe on electron microscope and light microscope. What are you going to write? Differences of specimen, right? Electron microscope and light microscope. You can see that the electron microscope, differences of the specimen. Live, no, we'll say dead, ne? Electron microscope dead, light microscope live. Dead specimen in EM, live or dead in LM. Then cannot see actual colors, ne? Can't see actual colors of specimen, but actual colors visible in light microscope. What does it going to say? Specimen can be directly observed. Can't observe directly. The other one can observe directly, means you are focusing light into your eye, ne? That you can't do with the electron microscope. What is the practical magnification of an electron microscope? What did we say, people? 500,000 times, ne? 500,000 times. Right, we come to the essay question. Describe regulation mechanism of enzyme action. People, you know, enzyme regulation 3. Enzyme regulation, what is that? Allosteric regulation you have to describe. Then cooperativity you have to describe. Feedback regulation you have to describe. All right. So this is the scheme. Right. This is the scheme. Okay. So we have to give a small description on that. I think you can take a screenshot. Ne? You can take a screenshot, and then this information, it's given in the resource, so you have to follow that. Okay, people. So did you take a screenshot? Right, check against your answer. So these points must be there. Right. So where is it? What is it? Where is it means what? You can't see the answer. Why? You can't see anything. Wait, why? Why that happened? Oh, okay. I shared the screen. No? Now can you see that? Can you see it now? Can or cannot? Can. All right. So now I'll see you. And thank you for joining tonight. See you soon. Bye.